Okay. Wow.
Hey everybody, welcome to Q&A with A&V. I'm Vincent Racaniello, and joining me tonight from New York, Amy Rosenfeld.
Hello,
Hey, Amy.
Vincent. How are you?
I'm well.
Do you want to tell them your exciting news? That your vacuum cleaner came?
Before we start, hello Frank.
Hello.
Thank you for returning and moderating.
Tell them where you were yesterday
Here's
without the
one more.
vacuum cleaner.
We all love Amy. There you go.
Thank you. But tell them without the vacuum cleaner.
Here's the story. Last week, someone said on the live stream, you need to be on Lex Friedman's podcast. I said, okay, tell him. The next day I got an email from him inviting me to Austin. And Tuesday I flew down, did a 4-hour pod with him. Just for proof, there he is. Okay? We'll have to take Amy off here, sorry. That's Lex, that's me. And so there you have it, folks. You wanted me to do it. I did it. Okay?
And it will be released when?
I don't know. I have no idea. I didn't ask. It was 4 hours, as I said, and that is a lot of time. But we had a great conversation, and I have to tell you something, folks. While we were recording, he gets a phone call, and he said, I have to take this, and it's Joe Rogan. They're buddies. So he chats with Rogan. He said, I'm recording with this guy, Vincent Racaniello. Oh, my God. Come on. And he has this podcast, This Week in Virology. You know about it? He said, no, I could hear him all the way across the room. His voice is so loud. So I'm thinking I could go on Joe. I could work Lex to get me on Joe. I know a lot of you don't like that. But, you know, Rogan is not vaccinated. He's worried about long-term effects. Blah, blah, blah. Anyway, okay, folks, that's the news of the week. I just got back today from Austin. I had dinner Tuesday night with Rich Condit, where I learned that Texas chili doesn't have any beans in it. I didn't know that.
I knew way before you did. I grew up knowing that.
I didn't know that. I tasted it. It was pretty good. I had a enchilada. We went to the Texas Chili Parlor, which, interestingly, was right next to the governor's mansion. Inside
so you had
of which,
a side of SARS-CoV-2 for dessert.
Inside of which was the governor with his positive COVID test after being vaccinated. They also gave him monoclonals.
Yeah, I know. And remdesivir, I think. But I might be wrong.
And all he was PCR positive. He had no symptoms. I think that's crazy. So that's the news for the week.
So when are you going to go use the vacuum cleaner? Most importantly is the vacuum cleaner.
Well, I think after TWIV on Friday, I'll go use the vacuum. I'll go and do more stuff. There's a lot of stuff here that has to be done. Before we take questions, there one issue I wanted to just chat about for 5 minutes. And this week, the narrative, there's always a narrative every bloody week, right? The narrative this week is boosters. The CDC and White House, not White House, but Fauci and the CDC are pushing we need boosters. So I've been doing a lot of reading. You know what? We don't need boosters. We don't need boosters. Let me show you a couple of papers just released today by CDC, which they say claims you need boosters. This one here, they looked at nursing home residents, effectiveness of the mRNA vaccines before and after circulation of the Delta variant. So the bottom line here is that effectiveness of the vaccines that's declined from 74 to 53%. But whenever you see the word effective, folks, what do? You ask, what are you talking about? And what they're talking about in this paper, if we scroll down here, it's just laboratory-confirmed infection.
No, it's laboratory-confirmed PCR.
PCR, it's not even infection.
Of RNA. It's just presence of RNA. You can't say it's infectious.
And they don't even know what kind of disease they have. Let's see here where it says this. This analysis assessed vaccine efficacy without being able to distinguish between asymptomatic and symptomatic. So We don't know if any of these people were sick. Okay, fine. That's Article 1. Here's Article 2. COVID cases in New York from May to July, okay? What happened? During May 3rd to July, vaccine effectiveness against hospitalization, here in New York, they know how to do this, was relatively stable. Really, the vaccine prevents you from being hospitalized. So in other words, we don't care if you get infected, which Amy and I have been saying for ages. And then one more. Here is a U.S. study, again, March to July, mRNA vaccines. Vaccine effectiveness, 86%, and against hospitalization again. 86% at 2 to 12 weeks and 84% at 13 to 24 weeks. So the effectiveness remains. Now, this is a 2% decline, which I think is probably not statistically significant. But the Surgeon General said today, we have evidence that the effectiveness against disease is declining. And I think that's what he was referring to.
2% is a calculation error.
It's ridiculous. So, folks.
Wait, what about the paper that I sent you earlier today, too? That talks about the year-long thing. I think it was a science paper. I sent you some papers.
Yes, there were multiple. There are multiple papers that also show that efficacy against disease stays the same. The bottom line is I see no evidence that we need a booster at this time.
No, they're just measuring antibodies, and antibodies are going to always decline. You want them to decline. Otherwise, you have too high an antibody count, and you get like autoimmune disease or whatever. You get bad things happen. Now, the question is whether or not, when you get reinfected in a year, your immune response clears the virus with either mild or no symptoms, right? Or you don't even get really infected. That's really the question. And you can't do that experiment till now because you're not a year away. I mean, a year is a year. It's like...
So what they're doing is an amateur thing. They are forgetting that for all vaccines, antibody levels decline after vaccination. For
everything. It's not
for
a
everything. For everything.
It's not a vaccine. The problem is now the administration has come out and said that if you are a nursing home and you don't vaccinate your residents and healthcare workers, you have the jeopardy of losing federal funding. So at this point, you might as well just vaccinate. Yeah. It's a Wall Street Journal article. At this point, you might as well just vaccinate.
That's like Florida saying, if you have a mask mandate, we're going to take away your funding. That's really the same thing. I'm very disappointed.
Yeah, I'm very disappointed. And I'm very disappointed. So while you were away, my godson's sister had someone in her class in front of her, directly in front of her, was tested positive. And so he wasn't in school since Wednesday. They sent the letter home, I think, on Monday, because grandma called me on Tuesday to ask what to do about whether or not the girl was going to come down with COVID, even though the girl and the kid were wearing masks, and whether or not they should get tested. And so my assessment was the girl was probably fine, but if they wanted a test for mine's sake, go ahead, get a test today or Thursday. And I confirmed my advice with Daniel. But
look,
you can't,
folks, you don't control infections by boosting people over and over, because the boost is going to decline within 6 to 8 months anyway. It's useless. It's useless. What you need to do is vaccinate more people. And by the way, I've focused on the U.S. Any country who says we need a booster based on infection data is wrong.
Well, it's Germany and it's Israel, and it's all really started with Israelis not understanding the right data. They didn't interpret their data properly, and they wrote papers, and then they were picked up, and because they're like a country of 9.5 million, and they could vaccinate almost everybody in the country, so it's kind of like a live in time experiment. It's just that it's unfortunate that they interpreted their data wrong.
So let's go on to some other things.
And they started, but you do realize that they started this problem when they came out and said, "Oh, look, vaccines prevent transmission because here, after 14 days of vaccination, we can't find any virus." You do realize that, right?
It was early after vaccination, right?
Yeah, it was way too early, and when the antibodies were just increasing.
Yeah. No, no. So the problem here is that everyone's forgotten their immunology 101. It's really sad.
Well, it's that, and the other thing is that they don't want to. A year is 365 days. That's how long it is. It's not going to be any shorter. It's not going to be any longer. And it's like when I used to go to the Derby, and they would say it's the fastest 2 minutes, and I would say to the people I was with, "Fastest 2 minutes? 2 minutes is 2 minutes. What do you mean it's the fastest 2 minutes?"
So someone said Booster Schmooster. That could be a TWIV title.
Yeah, you could use it tomorrow.
So let me welcome Vanity Nutrition also. Thank you for coming back. Appreciate it. All right, on to the on to the questions. Osterholm wants kids to wear KN95s. I don't care what Osterholm wants. Let the schools decide
Let the
what they
parents
have.
buy the masks and put them
Yes.
on the kids. And whatever mask your kid is willing to wear, I'm willing to accept.
Yep. And I remind you folks, if you want to ask us a question, put at Vincent Racaniello or at Amy. What is your YouTube handle, Amy? You don't even
YouTube
know.
handle? I don't
Just put
know.
at Amy. Just put at Amy. Right?
Yeah, I don't have YouTube handle. Really? I don't need more crap.
Okay. The problem is that many of the questions that were posted before the stream actually began are gone on my end. So please repost them. There are a lot of really good questions, folks. I'm really sorry about it, and I can't go and select them and answer them. Like.
But I didn't
So,
realize you were selecting and answering. So what am I doing here? I'm comic relief?
the question goes up. You're not comic relief. The question goes up. You look at it, and we both answer it, or mostly you answer it.
Not
But
most
someone
days.
wanted an update on the studio. I haven't been there this week because I was in Austin, so I'm going to go Friday, as I said. There you
Yeah, that
go.
was the whole joke about the vacuum cleaner.
Yeah, the All About R article in Nature mentioned in the pod was great. It's a really good article, yeah. I have a better understanding of R myself. All right, Elena. I'm a high school science teacher expecting to hear from our district that we will be in person with masks. Fine by me, and I always open a window even in winter months. It's perfect. That's the right thing to do. Did I say I was? Yes, I said I was in Austin, right?
Yeah, we talked about Austin. And the chili.
Do we know why we see mutations at the N-terminal domain?
Well, the NTD is an neutralizing antigenic site.
And
We don't know how it functions as a neutralizing antigenic site, but that's a detail.
Last TWIV led me to conclude that testing unless daily is pretty useless and we should go vaccinate only. Comment. Now, testing is not useless. It depends what your goal is. If you feel ill and you want to know if you have COVID, say you haven't been vaccinated, then you should get a test. You should get a PCR test, and if you're positive, you should isolate yourself for a while. That's a PCR test, a nucleic acid test. An antigen test has a different use. Now, we don't have cheap ones that you could do every day, but you could also test yourself with an antigen test. There are self-administered antigen tests that you could use as well. Thoughts, Amy?
I didn't realize that that's what he was talking about.
He said testing is useless unless it's daily.
Well, is he talking about testing vaccinated people, or is he talking about testing unvaccinated people so that they know what their status is?
So we talked on TWIV about testing vaccinated people, which you would say is not useful, right?
I think if you are doing a scientific experiment about how vaccination works and stuff, it's useful. I think if you're doing it just to do, I'm not really clear what its value is. Although I did listen to the woman who is survivors or whatever, the long-term COVID thing. She has the long-term COVID. She was in the bubble, and she's like, you should test because most depending on your age, if you're mildly symptomatic, you have the potential for long term. You have a greater potential for long term COVID. Not really clear what the relationship to that is with vaccination.
No, we don't
Um,
know. We don't
right.
know that at all.
But I think that we're overreacting when you get a positive test from vaccination, because we know that if after you're vaccinated, your RNA levels drop precipitously
Mm-hmm.
within like a day and a half.
Right.
So I'm not even clear that that is at the end, uh, ever leads to an infection, like a productive infection where you make productive progeny.
Yeah. All right. From EDA, my mom, vaccinated, will visit from another country, and I have unvaccinated toddlers. How long should she quarantine? What is the accuracy and best timing for the Abbott at-home test? Well,
Well, I
what
don't
would
have a
you?
problem with her being. I don't have her problem with her be. Why does she need to quarantine if she's vaccinated?
Yeah, well, I think EDA is assuming that if she got infected, she might transmit. That's the problem. So you could.
Well, it's not clear to me that you transmit.
No, it's not at all clear. So if you want to be safe, have her mask, and
Yeah, I
then
don't think she needs to quarantine.
doesn't need to quarantine. No,
Um,
don't
what? Don't what?
don't waste your precious time with your toddlers, right?
Right. I mean, grandma only comes once, right?
Yep. I am 13 and I would want to learn more about virology. Do you know any good resources where I can learn for free?
You go to you go to Prof VRR at YouTube and you download all of his lectures from 2021. Call it a day.
I don't know, 13? Do you think the 13-year-old could understand those lectures?
Yes.
We have
They
had
know what
over.
RNA is. They know what DNA is. They know what translation is
You
and
could try them.
transcription.
Yeah, on this YouTube where you are, you could start with there. There's some books if you'd like.
No, just
With
listen to the lectures and call it a day.
okay. Thank you. I saw a lot of people on the plane wearing 95s. What's the best way to keep kids safe? Everyone around her is vaccinated. Not everyone around us, especially in stores where masks are not required. All vaccinated 5 months now.
Wear a mask.
I think you should wear a mask. Yep.
I know, like, Daniel has the 80-20 rule or suggestion, but I think, it's kind of like a door. Goes, controls what goes in, controls what goes out.
Yeah. Okay. Here's one from Trevor. Third dose boosters are not needed. Well, we just talked about that, right?
Yeah. No,
Yeah, we have
Delta's
a lot of
not changing it.
booster questions. Here's from Mark. I think we got enough out on boosters, don't you think? Yeah. Yeah, we can. We're not boostering up. Absolutely not. I see no science saying that you need a booster. No science whatsoever. Right, Amy?
Yeah, for
Are you
sure.
getting a booster, Amy? No.
No, but, somebody like grandma called and my mom called and they all have booster appointments. And I'm a little confused about, well, my mom technically could be immune compromised, right?
Yes, sure.
But I'm a little confused even by boostering if you're immune compromised, if you don't know, because immune compromised could be anything, right? But
Yeah.
if you're immune compromised in B cell deficiencies, then I'm unclear what the boosters going to tell why you're getting a booster, right?
Boosters
Because
makes no sense.
it's going to make no it's not going to help you because you don't have any B cells to make any antibodies. Now, if you were worried about the T cells, then you should develop some kind of vaccine specific T cell test, right?
Yep.
Because I understand that the T cell test that's available really looks at nucleocapsid, so only is
Natural infection.
yeah.
All right. Is there a trial for the tweaking of the vax to work on Delta? the only trial I know of is, and this has been partly published, is Moderna did a South African variant. What is that? Beta?
I think that's gamma. Oh, it's beta. It's
Beta.
not gamma.
Maybe gamma. I don't remember. But South African 351, they modified the spike to include changes in that one, and that's underway. But that's not the one you'd want, right? You'd want Delta.
Well, there is right, but there's no need to change the vaccine because Theodora's papers have demonstrated that the antibodies that they collect are neutralizing against all variants, even though the vaccine that the people got was against the, what are we calling it now?
Ancestral
virus.
Pandemic has been a stress test of our healthcare system. Besides therapeutics, what do you think are other weak points that need to be patched? Science, policy. Go ahead, Amy.
All of the above and 400 others.
What else?
We need to reassess. We need science. We need policy. We need to understand the economic impact. We need to understand the social impact to children and parents and schooling. We need to reassess whether or not how we define risky experiments and not risky experiments for the NIH to fund. We need to reassess the whole kit and caboodle.
Lex Friedman had two other things. He said we should be better prepared to make new diagnostics if a new virus emerges.
Yeah, but that's just that's a repeat of Laurie Garrett.
And then he said masks. He said we should really figure out the best masks, do the right experiments, and have them ready. Yeah, I think that go ahead.
Laurie Garrett was all about the diagnostic. Yeah. He just took it from her. She really did not agree with you where you said that you thought it was vaccine and antiviral. She's like, it's all about diagnostic. But then what is your diagnostic test? Is your diagnostic test looking for the genetic material of the pathogen, or is your diagnostic test to actually look whether or not infectious pathogen is in you?
You want to see my diagnostic test?
Sure. Okay.
What's the guy's name? The Great something?
The Great
Karnak,
right?
I also think communication.
Which you just told me.
What?
Yeah. But you just told me. Go ahead.
Communication is a big flaw, too, because between the scientists and the journalists and the writers and the news people, I mean, I sat in the airport this morning and had CNN blaring with people saying idiotic things. I just don't get it. And they have so-called experts who don't know what they're talking about. And they had me on CNN over a year ago, and they never had me back because I said the truth, I guess, and they just want
you said things that they don't. You said what did I write to you this morning while you were watching CNN? It's the wrong answer or the wrong right answer. It's not the answer that they want to hear.
Will Shane Crotty be on TWIV? I'm thinking about it. Yeah, it would be fun.
To talk about what?
Well,
to talk about what exactly?
He wants to know about a third dose, but we already know about the third dose.
Right. So what exactly are you going to talk to Shane about? What's the question?
That's why he's not on yet. But Friday, Theodora and Paul are coming back to talk about their polymutant.
Yeah, I would actually like. I think their paper that says that natural infection leads to better antibody response and bigger breadth of antibodies against other variants is actually more interesting because it's the complete opposite of what Daniel has said, which is that vaccination leads to greater immunity or protection.
Do we have it in our folder?
Yeah, it's in the folder.
All right, we can talk about that too. We can talk about multiple things. All right, Bill wants to know, what's your thoughts on why we are not updating the mRNA vaccines before suggesting everyone get a booster? Won't we just be in the same situation 6 months from now?
We are not updating the mRNA vaccines because I believe you have to do a short clinical trial, right? And there hasn't been
Yeah.
any time. And they're rushing because of politics. And yes, every time you go and you do a booster, you will be in the same position 6 to 8 months later because all you're doing is quantifying the spike of antibodies that are induced by the booster. You're not actually understanding anything else. And really, what you need to do is you need to go out the whole year and 15 months and then ask whether or not cells from those people respond to virus as efficiently as they did at the beginning, right?
And also, Amy, do you remember when they took people who they gave some people a third dose a while ago, and it didn't make any difference beyond the second dose in terms of
Right,
breadth and durability. So
right.
I
That's
don't
what I'm
think it's going to
saying.
have any effect. Yeah.
That's what I'm saying. And the other thing is I'm not really, it's more important to understand how to induce the memory T and B cell responses and whether or not how long those last.
Yeah. Can you explain the dosing difference with Pfizer and Moderna? Do you remember how many micrograms of RNA
I
in one
think
or the
it's
other?
like 50 and 100.
And then
And
Moderna is a week later for the second dose, right?
Pfizer, yeah, it's a week later for the second dose.
Yeah.
I mean, it's really not there is really no explanation for the difference. I mean, there's not really a difference. A week and twice as much is not really a difference. I heard Moderna has 2 more doses than Pfizer. No, they're both 2-dose
And about 2
vaccines.
doses. Yeah.
So no.
It's a Dyson, Mark. Amy
Oh,
said I have to
those
get a
are
Dyson.
those are vacuum cleaner brands.
Yeah, they're famous old vacuum cleaner brands.
Well, what do I know? I'm not so good with the house.
CDC is predicting people with double shots will start to be hospitalized or want to quit. What prediction is not what we need right now. Prediction based on no data. Sorry, CDC.
Prediction? So did they get out their 8 ball? Because I'd like to predict that I'm going to get a job in the next 6 months.
This is how they predicted it.
Oh, can you tell me if I'm going to get the 2 jobs that I'm going to apply for in the next 6 months?
You will get one of the two. The great
Okay.
Karnak has spoken.
Okay. Well, that's good. That's good. Will it be the one I want or the one that has better weather? Actually,
Karnak only has so many powers. I'm sorry.
I was going to say, actually, I want both, but I only want to live in one of those places because the other place does not have as good weather.
So, Patricia, it was you who said I needed to be on. Well, you got what you asked for, okay? And he interviewed Minna last year, I think, after we had interviewed him. So Lex tells me he listens to Twiv.
Well,
I'm not
that's
sure
good
because
then.
he couldn't pronounce my name, so I don't know,
Well, that's irrelevant. But if he listens to Twiv and he's watched your lectures, he might want to go back and review. He's got a few factoids wrong.
4 hours is a long dog walk. You could split it up. And the cool thing is that he does this in his kitchen. His kitchen has no food in it, as far as I could tell. It's just got
Maybe
recording.
he has a second kitchen on the top 2 floors of the thing. Where does the guy eat? He obviously eats.
The kitchen seems to have a tea maker and a recording equipment on the center. There's a center island counter. It's covered with microphones and tripods and wires and stuff, which to me is awesome. I love it, right? But you know what? I said, this is really cool. And he said, that's because I live alone. Because if you live with someone, you can't leave recording equipment all over the counter. Right?
Why? You leave recording equipment all over.
No, I don't. It's all here in my office. It's here in my office.
Have you seen your office here?
My office
Have you
is full of
seen my
recording
lab? You have a recording equipment. I have lights and other stuff.
Anyway, it was a good.
What was in the refrigerator?
What, his refrigerator?
Do you have a refrigerator?
I guess he did, and he at one point he opened it. It was almost empty, and he had cans of energy drink, right? That young people drink to keep awake.
Oh, like Red Bull?
Yeah, but the one with the 3
I
claw
don't know.
marks on it. I don't know what it is. We're old
I'm
folk.
not. Hey, speak for yourself.
Yeah, you're right. I had a good
I'm not
time.
that old.
I had a good time. He's going to come to New York and visit the incubator, and I'm going to interview him about AI and robots because I'm really interested. And he's starting a robot company. He wants to put a robot in everybody's home.
Isn't that Alexa?
Amy, what would be the name of your robot?
The name of my robot?
Yeah, what would you call it?
I don't know. Maybe Phoebe? I don't know.
Yes, Mark Lex was born in Russia. He's lived in Moscow and has zero accent though. But he was very nice, very interesting, very interested, and he enjoyed the conversation very much. He thought it was amazing. So yeah, he doesn't know rock and yellow. What is it? Black and yellow, or rock and yellow, or raccoon yellow. No, he doesn't know any of that stuff. I'm sorry, James. I'm sorry. I get like 50 emails a day. I'm really sorry. And some days we don't get to email, but we'll get to it, James.
Well, you'll put it on the end for Friday. You'll make it a Friday thing. This Friday with after Paul and Theodore.
Well, I'm not sure
Or
that
they'll Paul and Theodore stay in the full 2 hours like they did last time.
I don't know. It's a good question.
And when are they going to Lake George? Are they going to go see Glenn?
They're going next week. That's why they're doing it this Friday, because they're going away next week. A key advantage of mRNA vaccines is the ability to keep up with changes. Right, yet boosters wouldn't even have D614G, never mind Delta. Well, you could change it, but as Amy said, we'd have to
You have to do
test
some
it. You
kind
have to
of
test it.
Yeah, the truth of the matter is you don't really need to change it because of Paul and Theodore's work.
Yeah. Ralph asked, did Lex ask about the meaning of life? Yeah, of course.
So what is the meaning of life?
I said it has no meaning. It's a random biochemical event that just happened. And it's just gone to amazing ends, but it has no meaning. No. So your life is what you make it. That's all. I really do think that. It has no meaning.
I don't really like the word meaning in the question.
It was his last question, but then I kept talking, and so it wasn't really his last question. Shocking. And I said, you better include all of this. Don't cut anything out. He said, no, 100% in.
Can't be 100% in, but okay.
Joe is not against vaccines. That's what Lex said, but he didn't get the COVID vaccine because he's worried about long-term effects. Look,
how old is Joe?
I don't know. I told Lex, look, if he wants the real scoop, tell him to get me on. But I said he'll probably be bored because I'm not going to mess around, okay? I'm just going to be serious.
Well, what I want to know is how old he is. Because if you're 50 and you're worried about long-term effects occurring in 50 years, you're going to be 100. And there are very few people who are 100 who don't have any aging effects. So I'm not clear how you're going to discern what is induced by the vaccine and what is induced by age.
If you like the Lex, it would be great to see you on Joe. I just think Joe would have no patience for me because, all right, so Lex was on the Rogan podcast the night before I was, right? That was Monday. And I said, what did you talk about? He said, we just drank whiskey and shot the shit. I'm not going to do that.
Well, that's what he does with Lex because they're buddies. But he might not do that with other people that he invites, like, I don't know, whoever else he has on his podcasts. I don't know. Who else has been on his podcast? I'm sure that there's a list.
Tons of people.
Wait, right, who are we discussing?
You're talking about Rogan, but I just want to get back to questions here. I have both Moderna, and I'm going by its timeline as 8 months since I didn't get it until April. I'm expecting a November BJAP.
Why?
You can get whatever you want. You don't need it. I'm not. I got it in February, March. I'm not getting a booster, folks.
No need. The problem is it's, I mean, so like Andy Slavitz came out and calculated, and it's like 200 million in the United States to give boosters is like 0.02% of the amount of billions of vaccines that we need to vaccinate the world. So it really, like, people who are arguing like, oh, it takes away from the world. It's going to inhibit the world. It's really not going to inhibit the world, but it's just the fact that we're so selfish.
So I wrote a blog post last week about the T cells. They're really what is responsible for protection after the first dose of vaccine when you don't see any neutralizing antibodies, and they persist. And as antibodies wane, neutralizing antibodies, the T cells are what protects you. And so that's Shane Crowdy's not worried. Yes, that's good. That's
The
really
truth
good.
is the vaccine is for T cells. It's not for B cells and antibodies. It takes too long. Vaccine is for inducing memory T cells.
Some people are hospitalized after and do get disease. Yes, it's never 100%. Would a third vax do any harm? How do you know which people are going to end up getting sick? You're going to just third boost everyone? I don't think that's a viable strategy, especially because there's a small fraction of people who go to the hospital who are double vaccinated.
Well, you also don't even know that those people responded to the first vaccine, to the first shots.
Yeah. Ralph is curious about mRNA after J&J. What are you curious about, Ralph? You got one J&J dose.
This is Angie. This is what Angie did. Is she got the J&J dose, and then she decided she needed a booster, and so she went and got Moderna, and she tweeted about it.
Yeah, and where's the science behind that? I'm waiting.
Oh. Yeah, that would be my response, too.
Okay, silence.
She kind of she took some slack for it.
She said she topped it up, and I think that's offensive to call it topping up. But let's move on.
Is that like what you do with the gas?
Yeah, you top up your tank.
Yeah, I was gonna say. And then don't you top up your beer when they pour you a draft beer? Like after they get rid of the head, then they like top it off or something. Yeah. I mean, I'm not a beer drinker, so I don't really know, but I think that's what
Thanks, BJR2, for your contribution to the incubator, the future of good science communication. And thank you, Axis Audio. Didn't Dr. Griffin say the data isn't there for immunocompromised? That's what he said last week. He feels the science is not there to say we need to give anyone a boost, including the immunocompromised who are getting them, apparently.
Yes.
No, so this is good. Data. Why not? Why only one? Surely we could get a 10% cash discount from Pfizer and Moderna for a monthly booster. Yeah, you could subscribe to the booster. That's perfect. Like your software. You don't buy software anymore. You subscribe to it. Subscribe to the booster. Which one are you going to subscribe to, Amy?
Well, the thing is I don't really like subscriptions because I don't always use all of my time on the subscription. So I'll get back to you.
I live in Florida, the epicenter of stupid. I'm sorry, overjoyed.
Yeah, my godson and his grandmother and his mother and his sister live in the epicenter of stupid, too. That's who called the other day. Sorry. You guys should vote him out. You have the opportunity to vote out stupid.
Preventing long COVID. By what? A booster? There's no evidence that it prevents long COVID. No.
That's what the lady says.
What lady?
The survivorclub.com or whatever that.
There's no evidence that a booster will do anything. We don't know anything about vaccination and long COVID right now. The study out of Israel stating the vaccine effectiveness wanes at 8 months. Infection, not disease. When you talk about effectiveness, you got to define what you're talking about. It wanes at preventing infection, but you're still protected against severe disease. Oi. Thank you, Ditching. Really appreciate your support. Are the boosters going to be different?
Really? Okay. Really, Frank? Really? You've been sitting here the entire hour. Really?
Maybe he posted this before we started talking.
He needs to, try asking a different question.
Well,
I'm done with this.
It is a question whether you're going to get the same thing that you got before or something different.
Yeah, that's what they're saying is you should get the same one as you got before.
All right.
Okay. Really?
Another narrative out there is vaccine waning, which has got a life of its own. This is totally ridiculous. There's no vaccine waning. You have memory.
Mean?
You have memory.
What does vaccine waning mean? Vaccines can't wane. You get it, and then
Vaccine
it goes
waning
away.
is, I don't want to go to Miami this winter. Oh, that's whining. I forgot.
That's whining.
I don't know.
I don't know what vaccine waning is because you
All these
get it, and
narratives. Jenster, most of the narratives are
what
not
is up with
good.
this word narrative?
I
It's not
like
a
the word.
narrative. Yeah, I don't. It's not a
Oh,
narrative.
6 months after I started using it, now you're complaining?
I've been actually. I complained when we wrote the editorial too. I wanted to take it out. I don't know what the fucking narrative is. This isn't this. It isn't fiction. Although I do have to say, my thesis reader might say to me, "Amy, you got to put in some bullshit in your discussion."
James, first of all, James, don't write in all caps, okay? It's offensive. And secondly, what people care about. People, if you save their life, that's all that matters. That's what vaccines do. They save your life. They prevent serious disease. There's no need to prevent infection. It's impossible, James, okay? More Delta boosters.
No.
All right, let's move on. Oh, here in New Zealand, our prime minister was called nuts by many in the world after locking down after one case. Now there are 10, maybe 50. What do you think? Guess the
Well,
lockdown didn't work. It's too late because when at the time of lockdown, there are already many other infections, right, Amy?
yeah, like it's a not successful. It's a non-successful policy. It was non-successful beforehand. This is the only time I would advocate where you should test your entire population every day. Well, it's not the only time. It was just an exaggeration. But this is where you should test your population every day.
How long does efficacy remain? Ian, I'm going to let you rephrase the question. I want you to tell me efficacy against what? All COVID, mild, asymptomatic, moderate, severe,
I
death.
don't think efficacy is the right word because efficacy
Oh,
is a percentage.
well,
So I think
efficacy is during the vaccine trial. Real
well,
world is effectiveness.
right, but it's a percentage. They're measuring
Yeah,
efficacy by 95%
yeah, that's right.
didn't do this. It's not how long suggests a temporal regulation. That's not the right way to. That's not the right. Your your question is confusing because it's two very different thoughts trying to combine to one.
How long are T cell responses likely to last? So I read a paper,
Years.
yeah, years, maybe decades. It's for much longer than, well,
B cells,
B cells.
antibodies, and that paper was a year. They looked at a year.
But PAMs can have long COVID or other damage, so isn't a third shot the precautionary principle? We don't know if vaccination still lets you get long COVID when you get PCR positive. We just don't know.
PCR positive is not what is PAM? Pre-symptomatic, asymptomatic, and mild
Yeah,
disease. What's
yeah.
the S?
Mildly symptomatic, moderately symptomatic, I think.
No. Oh,
Pre-symptomatic, asymptomatic, and mildly symptomatic.
so yeah. I mean, the
But
third
Charles,
shot is not going to prevent that. It's not a
Charles,
precautionary principle because you
and,
still get infection.
and let's say it did. So you're going to give everyone a booster every 8 months? Really?
Well,
That's a strategy?
you don't need a booster every 8 months. That's the problem. Is that you didn't go out long enough. It's just like the whole discussion that we had about
Yeah.
whether or not you should have the second boost, the first, whatever, the second shot 4 weeks after the first shot, which is why I got so upset about the person who then came and said, "Oh, it's because the response decays," and it's like, no, the response doesn't get decay in 4 weeks. In 4 weeks. Remember that?
Mm-hmm.
And then you said to me the other day, "Oh, she made one mistake," and it was like, no, it's a whole big principle of a mistake, right?
Right.
So this is the same mistake. is you haven't gone out long enough to really assess what immunity, how long immunity lasts. You went and you said, "Oh, look, there's a decay of antibodies," without recognizing that that's what you want. And so again, it's premature to make any decision because it you need at least a year.
So why not get a booster? Because it takes vax away? What's the downside? I think this is funny because we have people who don't want to get any vaccine, and now we want people who want boosters. It's like, isn't that odd? Anyway, what's the
No,
downside? All right,
it's not
Mark,
odd.
I'm just saying I want things done based on science. Why not is not an excuse to get a booster. What's the downside? You don't need it. That's the downside. Morris, thank you for your contribution. How much less is immunity for folks who took shots 3 weeks apart as opposed to 9 to 12 weeks apart?
Immunity at what point? At what time point? Immunity 3 months after you got vaccinated?
So in the end,
Immunity.
I mean, so in the end, if you're 6 months out of both now, are you going to
No,
have it?
there's no difference.
There's no difference. No, that's what I
No,
thought.
there's no difference.
I think that if disease increases, then you think about another vaccine, but don't just give the same vaccine over and over. Make something different that deals with it. All right, here's one I don't know the answer to. Any idea why human papillomavirus vaccines appear near sterilizing?
Because they don't allow for infection.
Yeah, but why? Because I think the way that you get infected, of course, is by the cervical mucosa, the genital urinary mucosa, right? So
Probably
somehow,
a lot of IgA, I mean.
yeah, they maintain high levels of IgA, which is very unusual. And we don't understand the basis for that, but it's really phenomenal.
All right, it's 8:45.
My stepmom got her third jab today. Okay. Wow, it's happening quickly.
Yeah, my aunt is going, or maybe she's going Friday, and my mom is going Sunday.
Yeah.
But
COVID.
see, here's the difference. My mom is taking the anti-CD20 antibody. Her husband is 20 years older than she is. And there's no discussion about him getting a booster because she recognizes that he's fine. But because she's taking this B cell, what's supposed to deplete your B cells, she's afraid she's not going to be able to be immune or whatever. So while there's a flaw in that logic, also, I'm more
Mm,
accepting of that than I am of others. But you see how
yeah.
the difference is. Like she logically thought out her 90 whatever year old husband doesn't need a booster because he's fine. He's got antibodies and
Yeah.
blah blah blah. Right?
So look at this
See,
question. Do you think masking has kept kids, immuno experienced? Is this going to be a problem?
it's the thought.
Yeah, we don't
It's
have
the
any
thought.
evidence, though.
No, actually, we do. It's the hypothesis that people are using to explain the rapid rise in RSV and rhino and
Mm-hmm.
parainfluenza infections that we see once we came out of lockdown. Yeah,
No,
that's
but he's
the
saying, will the immune response be weaker? So that makes perfect sense that they haven't seen those viruses and now suddenly they're seeing them. But
weaker suggests that these weaker. I don't think that the question is phrased properly. Personally, I think what he's trying to ask is, did they have were they immunologically naive against those viruses, and are we going if we continue to mask and the hygiene hypothesis, will you continue to see
mm,
these outbreaks because you will be more of us will be immune naive.
all right, one more, okay?
Uh-huh.
Israel is stating they're having increased hospitalization, Pfizer, after 8 months. Is that the 2% that we saw here in the U.S., you think, Amy? Have you?
Probably.
Have you?
I
So
haven't seen anything like that,
let's see the
but
data.
probably.
But T Way, if that's true, that's what you need to say we need a booster. Increased hospitalization in fully vaccinated people. And as we said at the top of the stream, we don't see that yet here.
Well, the
But it's
other thing is just reporting increased hospitalization is not does not correlate with vaccine people. You cannot deduce that those increases are vaccinated people. And the lady who was the epidemiologist from Hopkins who Slavitt interviewed, she clearly states, like, yeah, we see an increase in hospitalizations, but those aren't the vaccinated people.
let me do one more.
And I think
Uh,
Danielle concurs, right?
yes. Are vaccines less effective for smokers?
Uh, I don't know. I would assume so.
You haven't seen the data, though, right?
I haven't seen any data.
Okay.
I haven't seen any data, but I would assume that, you know, just like you're more subjected to disease when you're a smoker.
Mm,
It's like a comorbidity. I would assume that there's some kind of side effect to
yeah.
the immune response, which makes you more susceptible to disease. So therefore, I would think that they would be less effective, but I haven't seen any data. And less effective compared to what? I mean, we don't really know what you need as a baseline level, right?
Also, effective against what? All COVID, severe, moderate?
Right,
There's a big
exactly.
difference.
Exactly.
Okay, Amy, thank you.
All right. Tomorrow, mice and more mice and
I'll
more
be there.
mice.
I'll be
All
there.
right. All right. Have a good
Thank you, Amy.
evening.
Good night.
Good night. Look, you're at 700.
Cool.
Yeah, it's good.
I didn't realize boosters would be such a thorny thing, controversial thing. Man, no data. You still want to take boosters. Let's see. Correlates of immunity. This is very interesting. I've been reading. There's a very nice paper where they tried to calculate a correlate, because a correlate meaning what do you need? Looks like antibodies are a correlate of protection of some kind. And so this one study calculated the correlates, and it's very interesting that so the amount of antibody in a convalescent serum, say, they did all standardization in this paper. The correlate of protection is about 20% of the level of neutralizing antibody that you would find in convalescent serum. What's interesting is that's for any COVID. The correlate for protection against severe disease and hospitalization is only 2% of the level of antibody, which means to me that T cells are what's doing that, because there's very little antibody needed for that. And going along with that, the same paper looked at the decay of serum antibody over time after vaccination. It decays about 2% a month. But the T cell protection doesn't decay. They think it lasts for decades, as we've seen before. We're gonna do a TWIV on that paper, probably next Friday, because this Friday we're having Paul and Theodora back. We had a friend who was vaccinated, caught COVID, was in the ICU. Should we have spaced out the booster by months, like in many other? No, because a certain number of months after the second dose, the booster of a normal 2 vaccine course, you have full-blown immunity. It's not going to get any better. But remember, it's unfortunate that he did. I sat next to someone on the plane today whose grandfather died. He was fully vaccinated. He was 95 years old with a lot of other comorbidities. Fully vaccinated, got COVID, died in the hospital. Highly unfortunate, but remember, they're not 100%. None of them are 100%. The CDC is giving everyone whiplash. Why don't they consult virologists? Well, there are a lot of virologists at CDC. I don't know how they formulate their policy. I would just like to see the basis for their decisions. Today, they say we're going to release 3 papers on MMWR that tell you why we think you need a booster. So I look at the 3 papers. They don't tell me why I need a booster. So you're hearing from me my interpretation. You can disagree. I don't care if you disagree or not, all right? You're absolutely entitled to do that, but I'm telling you why I think we don't need a booster. More questions. I'm looking for my name, and I don't see them here. Will booster shots increase variants? So I've heard this from a lot of people, including Lex. I think he asked me if any vaccination will select for variants. Any immunity will select for variants of any kind, of course. Now, some people say it will increase the mutation rate. That's wrong to say that. The immunity does not increase the mutation rate. The mutation rate of a virus is intrinsic to its RNA polymerase. It is not influenced by immunity. What does happen is the immunity selects for the rare variants that arise that have changes that, say, evade neutralization by antibody. So any kind of immunity, will select for variants that evade it, whether the immunity is given by natural infection or by vaccination. So many people take that to mean we shouldn't be vaccinating. Good luck with that, as Amy would say. Your choice is not to get vaccinated and risk dying or getting very severe disease, or getting vaccinated and having us deal with variants that arise. With influenza virus, we make a new vaccine every couple of years. Maybe we have to do that with COVID. But giving a booster of the same vaccine doesn't make any sense to me, for reasons that I've told you. Let's see, some of the questions. Any possible negative effect? Well, they've done some limited 3-dose vaccinations. Doesn't seem to be any negative effects, no. So I'm not arguing against it because of that. It's like ivermectin, say. Daniel and I don't see the evidence that it helps, but many people take it. If you want to take it, that's fine. If you want a 3rd dose, it's fine. I'm just telling you what I think about a 3rd dose. And more importantly, why the public health policy doesn't make sense, because it's not scientifically justified. Has TWIV ever done or can do a show on how RNA might have evolved to be the first carrier of genetic information? So the first the first thought I have is that a little out of focus here. Let's focus it up. Now it's even worse. First thought I have is that Eugene Koonin did this. Here we go. We had Eugene Koonin on a while ago. So Rich Condit and I went down to Bethesda to talk to him, and he talked a bit about that. So go check out that episode. It's the last Koonin episode. We've had 2. All right, Amy's gone, but I'll take her questions. Long COVID for 17 and a half months. Had antibodies on July, no antibodies in December. Second vaccine early is April. Last week, LabCorp antibodies 657. I don't want even a mild infection. Advice. Well, if you don't want to get any kind of COVID, mask up and distance. Daniel doesn't want any kind of COVID. He says he masks up, he stays away from large crowds and so forth. You have to really be careful. If you have to work, it's tough, but you need to wear a mask. And remember, the mask isn't 100%, so you have to do other things like distancing. Does sterilizing immunity even make sense? So, you can do an experiment, and that's what they did after the vaccines got their EUAs and were used widely. So remember, the vaccines were tested just by seeing if they prevent any kind of COVID. And then after they were used a lot, they said, okay, let's see if the vaccines prevent infection. And so they looked at people a month or 2 after vaccination. And what they do is they have you do a PCR every few days, a swab. You send the swab in. And then they see if you get PCR positive. And then they could figure out that early on it really did prevent infection when you have very high serum antibodies and high mucosal antibodies. But as time goes on, you lose about 2% of those antibodies every month. And eventually you get reinfected. And first, reported in Israel, everyone all up in arms, the vaccines don't work. Think, people, the vaccines do work. No, with the exception of the HPV vaccine, they all allow infection. This is the way it works. They prevent disease. So you could do a study and, to see. But anyway, for this vaccine, it's not sterilizing for more than a few months after you get the vaccine. And so, but vaccines are tested just to prevent disease. This was a study done afterwards to see if people would shed virus and so forth. And I think we're not going to have any of these vaccines be sterilizing, the COVID vaccines, that is. With the shorter incubation period of Delta, is that enough of a different effect to upgrade Delta from a variant to a strain? Well, I think you have to have some more data aside from the epidemiological data, which is done in some people but not enough. And we also need cell culture experiments to corroborate that. And then, as Jens Kuhn says, an international committee will look at it and decide if it's a strain. So far, it's a variant. For those that are immune compromised, does the booster help? Well, it's been tested in transplant recipients, and getting a third dose helps some of them, not all of them, and that's why those are some of the data that have been used to recommend boosters for immunosuppressed patients. Shane Karate, we already addressed him. Any news on antivirals? Nothing yet. I'm surprised that we haven't heard from molnupiravir, right, which is in phase 3. And, weeks ago, Daniel said we should hear any day now about it, but we have heard nothing. So I don't know. If you could hazard a guess, how concerned are you about a variant with higher mortality? I'm not concerned at all because... All right, so here's the scenario. Higher virulence, which is what you're talking about, is not really selected for in evolution. Now, it might not have to be selected for. It could just arise accidentally as a consequence of some other change that were selected. So you could imagine that a change in the virus that allowed it to evade some kind of immunity could also make it more virulent. But remember, SARS-1 it was sort of like that in that most of the patients were in the hospital when they were shedding, so they weren't able to transmit very effectively. And of course, also for SARS-1, there was no pre-symptomatic, asymptomatic shedding, and that contributed to it as well. My feeling is that if you're sicker, you're less likely to be moving around and transmitting, right? But that's really hypothetical. So I think it's unlikely. In all the virus outbreaks I've been looking at over the years, I haven't seen it happen, which is not to say it won't, but I think it's highly unlikely. How would you design a new flu vaccine versus the current one? Well, I'm going to be highly unoriginal here and just say I will make an mRNA for the hemagglutinin and maybe the neuraminidase and put it in a lipid nanoparticle and see how that does. I mean, it's not going to work for every vaccine, but that's pretty easy to try. What you really want, though, is a flu vaccine that we don't have to make again every few years. You want a so-called universal flu vaccine, right? And to do that, you need to induce antibodies against conserved epitopes on the hemagglutinin, on the spike. And people are trying that, and I think that's a good solution by taking the stem of the HA and somehow directing the immune response. The stem is immunosubdominant, which means that when you're infected with influenza virus, most of the antibodies against the HA against the head, not the stem. So we don't get a lot of these broadly neutralizing antibodies. But they're there, and they work really well to neutralize many different kinds of influenza viruses. So a strategy to induce those would be very interesting. How was your experience flying to Austin? So Newark Airport was totally jammed at 6 a.m. on Tuesday. It's ridiculous. But everyone was masked because it's a federal facility. You have to be masked, and if you're not, they come right up to you. Even the bus driver driving me from my parking lot. If someone walked in without a mask, the bus driver wouldn't move until they put their mask on. Everybody's masked. Everybody's on the plane. The plane was 100% packed, but everyone's masked up. So I think that's good. So it was fine. It's a 3-hour flight, and then today flying back again, the flight was jammed. People are on vacation. They're all wearing shorts. I'm the only guy with a suit, with a sports jacket, doing work, and everyone else is on vacation. But that's fine. So it was fine. And that was my first flight since February 2020. My super chat question: on what basis did Greg Abbott get MABS? I don't know. Oh, the governor of Texas. Sorry. I'm confusing Abbott with the company. Because he's over 60 and he's paralyzed, and so comorbidities, right? And he's a politician, so he can get whatever he wants or what his handlers think he should get. So I was going to ask Daniel tomorrow that if you had a 60-year-old patient who's fully vaccinated, tests PCR positive but has no symptoms, would you give him monoclonals and remdesivir? Frankly, I think it's overkill. But he's the governor of a state, so they want to protect it. Okay. Recent study proposed low blood sugar correlated to COVID-19 severity. I have never heard that. I would love to see the data. And I don't think maintaining high blood sugar is a good strategy, right? Because that can lead to other pathologies. There are other ways to prevent viral diseases, as you know. Did I see today's Bloomberg? Where did COVID come from? So I didn't, but I saw a science article which concluded that it came from Nature. It's basically the same data that have been rehashed over the past months. That it came from Nature, which of course I agree with, and there's no new, as far as I know, there's no new evidence, but someone is saying here, Bloomberg reported a paper that was found pointing at a zoological source. As I say, there's a review article. I don't think it's an actual data paper. What does the end of the pandemic look like? Well, it looks like the pandemic's not over till most of the world is vaccinated, right? And someone asked about herd immunity. Maybe I skipped that. I don't know. Anyway, the pandemic is when most of the world is vaccinated. Circulation slowly declines. However, there are going to be seasonal outbreaks. I think every winter there's going to be an outbreak, mainly in unvaccinated people. And when could that be? Well, seeing that many countries have hardly been vaccinated, I think it's going to be at least another year, because obviously production is not sufficient to vaccinate most of the world. And then you have countries like us where we're just over 50%. How do we get higher? We need to get higher. So I think it takes longer. I frankly thought it would be over this fall for the U.S. at least, because I thought we'd have 80% vaccination, but I was wrong. I was wrong about that. What's holding up funding on long COVID? Oh, no, the NIH got a bolus of money to do that. That's moving forward for sure. For sure. What are your thoughts on Adenovector vaccines? I think these are very good vaccines. And the J&J, Janssen, as you say, 100% preventing death and hospitalization in South Africa, which is a different virus from the one in the vector. So I think these are effective vaccines. Should they receive an mRNA booster? The data on those are not as comprehensive as for the mRNA vaccines in terms of continued protection against severe disease, right? So I would reserve a judgment on that. But obviously people are doing it anyway. Can you please explain why the lab leak is flawed? Okay, so here's my position. I don't know if you would say flawed, but it is a hypothesis without experimental data supporting it, right? Not experimental, any kind of data. There's no data. It's just a theory. It could be because, we first detected the virus in Wuhan. There's that Wuhan Institute of Virology that happens to work on SARS-like coronaviruses, right? Well, this coincidence. And so the reason I think it didn't come from the lab, first of all, nobody had anything like SARS-CoV-2 before December 2019. No one had it. And if you think they were hiding it, well, that's your idea, but that's not science. I go by the science. The head of the lab in the Wuhan Institute of Virology, Zhang Shi, said she wasn't working on the virus. She had nothing close to it. She didn't have any bats with it. And in fact, the closest virus is still over 1,000 bases different from SARS-CoV-2 that anybody has gotten from a bat. So nobody had a virus close enough to have released it. She said she wasn't working on them. She knew all the viruses she had in the laboratory. And you know what? I believe her. On the other hand, we have now SARS-CoV-2-like viruses being isolated, not only from China, but Southeast Asia, all the way up to Japan, from bats and pangolins. And so there are many potential sources. The SARS-1 came from a live meat market. It's possible that that was the source of this one. So I think all the wildlife data support a wildlife origin, as does all the previous outbreaks, spillovers of zoonotic pathogens that have come from animals. And none have come from a lab. And there's no evidence that she had it and could release it. So it doesn't make any sense to me. You could not believe her. That's fine, as I said. But did you see Ravi Gupta say Pfizer bribed labs to underplay data on Delta? No, but I think he or she was just messing around, and you don't do that because on Twitter nothing is peer reviewed, right? Yeah, you should open windows for sure. Good ventilation is great, and on an airplane there's a lot of air ventilation, right? It's great. If a vaccinated mom stops breastfeeding, does the child lose the IgA? Well, yes. The antibody, and of course it depends. The length of time it lasts depends on how long the child was breastfed. But even if you breastfeed for 6 months and stop, then the antibodies will only last a few months. They don't last forever. So at some point you always stop breastfeeding and then the antibodies decline. But by then, the idea is that the baby can make their own antibodies. Does boosting the immune system in conjunction with the jab have a better advantage than a booster shot? You mean like putting adjuvants in the vaccine? Well, I think the adjuvants do give you a higher affinity and a broader immune response, and the lipid nanoparticles, for example, are an example of an adjuvant that's with the vaccine. But advantage as far as what goes, though, what are you thinking of? Are you thinking about memory? Are you thinking about breadth of the antibody or T cell response? Also, the third shot doesn't really do much except boost your antibodies for a short amount of time. It doesn't broaden the antibody response. It doesn't give you more memory. It just gives you more serum antibodies so you won't get infected for 8 months maybe. I don't think that's a strategy going forward. I think the strategy is to vaccinate more people so that you can cut down the circulation. And you know what, folks? I mean, you may personally want a third boost because you think it might help, but that's fine. I don't think we're going to get a third boost into 50% of the U.S. I think a lot of those people have had it. They're not going to go back. And I think that's part of the problem here is that we're not thinking it through in terms of a PR situation. When was your first inkling that COVID would be such a huge deal? So I remember in January we heard the first report. We did our first TWIV, January 2020. I was teaching my virology class, and before every class I watched it spread. And, it spread really extensively in China, but not elsewhere. And I thought, oh, this is not going anywhere. And then all of a sudden, it's popping up all over the place. And then I said, oh, this is going to be a problem, just in terms of spread. But also, we knew that a lot of the infections, a number of infections in China that were initially picked up were fatal, right? So it could kill people. So I think probably March, because then our university shut down. And I said, whoa, this is a big problem. MMWR used to be an alert, not a validation for crap policies. What do you think about VAERS? Because there's thousands of cases related to fertility and COVID vaccine. Is the data meaningful if it's self-reported? Well, you have to go through all of it, delineate it, because, you have self-reporting issues and you have to sort through those. You also have things that happen, right? When you immunize millions of people, other things happen, obviously, and you have to separate those from the vaccine. So you have to look at the timing and just make a decision on whether it's vaccine related. And they go through all those cases. So I think it's useful because it can reveal issues that aren't picked up in other ways. But it is also misused, as you know. Does New York have an exposure notification app? Yes. Yes, they do. I have it, and Amy has it on the phone. What should Vaccinated do if they are pinged? If you're pinged as being near a person known to be positive, well, you're fully vaccinated. I think you're fine. But if you want to know if you're infected, you could do a self-antigen test if you'd like, or you could go get a PCR test. But as I said, it's not going to tell you if you're actually infected. What really is the issue is whether you get symptoms or not. So just to be safe, if you're around young kids, you could get tested. If you're positive, quarantine yourself for 10 days or so. Does COVID do anything, you're talking about SARS-CoV-2, the virus that causes COVID, to infected cells other than make more of itself? Oh, no, it kills the cells that it infects, for sure. That's a property we call cytotoxic, right? Cytotoxicity. Any cell that gets in, it kills it, yeah. And so enough cells get killed, it could cause some damage. But, in the upper tract, mainly the infected cells are shedding a ton of virus, and then you transmit it. There's a little bit of epithelial damage, but most of the symptoms are actually your immune response kicking in and making you make mucus and sneeze and cough and have sore throat and so forth. The damage to the cells has more of an impact in the lower tract, down deep in the lung, where it can damage the alveoli and make it difficult for the body to keep fluid out of the alveolar sacs, and you get pneumonia, and you can drown in your own fluids. Would a healthy 40-year-old who went on a course of steroids at the first dose more likely need a boost? It depends on the steroid and how much and how it was taken. But in some cases, Daniel has said, yes, if you were on steroids at the time of your vaccine, you should probably get another dose. Absolutely. But you can go back and listen to Daniel on that. Thoughts on the mechanism of action for Lin Fo Wang's paper, Pan-SARS-Becovirus Neutralizing Antibodies. So we did that paper on TWIV, and they're directed against a antigenic site, which is not the usual one that's been recognized by all the antibodies and monoclonals that have been made. It's a different site, and it's highly conserved among different viruses, and that's very interesting. And getting people to make those in response to vaccination is a goal. And that could avoid emergence of variants, right? If you could somehow induce those antibodies. This is the kind of thing that we don't learn until a long time into this pandemic because basic research is being done. And it's too late to make a vaccine with that because our vaccines are already done, right? But if we can figure out how to induce these broadly neutralizing antibodies, that's next generation vaccine. It gets around variant issues, right? Theoretically. So I hope somebody's pushing that forward. It'll be great. Did you get flack regarding your Times article on the variants? No flag. I think it was ignored because the narrative in the Times, at least, and other mass media is that the variants are more transmissible, and that's causing the problem. Where if you heard Jeff Shaman, he said a lot of things are problems besides the virus itself, and figuring out the virus's contribution is tough. But certainly people's behavior and so forth, and that was the message from our op-ed. But no flack. We got some people agreeing with us, but mainly I think it was ignored. All right, so you want me to invite an immunologist because you don't like my position on boosters. No problem. In fact, no one person knows everything for sure. But when policy is made without the data, and I can bring on Crotty, and if he says we need boosters, but he's not. He's not going to say that. If someone said we need boosters, I say, why do we need boosters? Well, they'll say the antibody levels are declining. I say, so what? You're still protected against severe disease. Then let's see what they say. Does PAX make sense post-acute sequelae, right? If long COVID can happen without symptomatic infection. So acute simply refers to a infection of a defined duration. It doesn't mean that you have symptoms, so it's fine, yes. Yep. Good question. Is there a way with vaccine to shorten the recall time of memory B cells? So that's the issue, right? If you're 12 months out of a vaccine, you now have very low serum and mucosal antibodies. So a virus enters your nose, it's going to start replicating, right? Because there are not enough antibodies around to stop it. But the memory response will kick in very quickly in 2 days maybe, as opposed to weeks if it were your first infection, okay? So that's the difference. You're going to make antibodies and T cells that are going to contain the virus. You may not even shed, and if you do, you might not shed enough to transmit. That's why herd immunity works, even though you get infected. So, can you shorten it? It's hardwired into your biology. I don't think you can do that. But I think it's worth thinking about, right? Let's know what the limitation is. I mean, here's what happens when the virus comes in. The antigen presenting cells like dendritic cells pick it up. They bring it into the lymph node where the T cells see it and start to proliferate, and the B cells and T cells start to proliferate. So, that's the delay. The lymph nodes are pretty close. They're down in your neck area, right? So that's pretty close. I don't know how you speed that up. You can't have the T cells up in your nasal passages. I mean, there are some there, but clearly there's still a delay. They're tissue resident memory T cells. So if there are enough there and it's just a matter of them ramping up, I don't see how you can do that. But it's a fascinating question. I really like that. All right, so I think we've answered this. Is there a downside on the individual level? Not that I know of. On a population level, I don't think we need it yet. We don't have the data for it. And I think more importantly, why are CDC saying we need it when there are no data to say? That disturbs me deeply. Okay, that's really what gets me about this. But if you want to get it, fine, you can go and get it. We get viruses from animals. Where do they get them? They're their viruses. Every animal on the planet and every living thing on the planet has its own collection of viruses, and tons of different ones, and making lots and lots of viruses that are mutating all the time and making variants that maybe now and then could infect a human if they ever contacted a human. And most animals never contact humans, most wild animals. But now and then they contact us. So the animals have had them forever, since they evolved. They inherited them from their ancestors and so forth. What happened to the lick-a-sticks? No funding. It's, I don't know. I should get Michael Minna on and explain that, right? Because that was a very exciting thing. It never happened. I think it's worth saying, Michael, what happened? Explain it to us, right? Do you want me to do that? I'll do it. You let me know. I went on Lex, boom, Let me know what you want me to do. I was talking about tests as an alternative to vaccination. So you test, and then if you're positive, you quarantine. Well, that's the idea of lick-a-stick, right? Before we had vaccines. So in theory, it works. But now we have vaccines, so why not use both, right? What should we tell our 85-year-old parents? Should we wait 1 year for boosters? They're both fully vaccinated. If they were my parents, I would not. 85-year-old parents, I would say you don't need a booster. But you have to make your own decision based on what you hear. Any reason why VE data from Moderna differs from Pfizer? I don't think they're that different, right? The one paper I'm looking at here, 86, 84, I think they're very similar. I don't think they differ. I could be wrong, but I think they're similar. Do we yet know if COVID reinfection is usually worse or less worse? It's usually less work. If you're vaccinated or a reinfection. You mean, if you get infected. Sorry, yes. You've said it in the question. I'm just stupid. It seems to be less worse. You do have good protection from natural infection, which is also durable. Yes, absolutely. Should the U.S. have delayed the second shot? No, I don't think so. The UK did that because of supply initially, right? And they had studies which suggested it would be okay. You don't get the full maturation of the antibody T-cell response until the second shot. You do get some protection, but it's not in the 90s after the first shot. So if we had a vaccine shortage, I would say, yeah, that would be fine to do that. Yep. How have you handled the emotional side of the pandemic? Now, this is a good question. I don't always handle it well. So I think I was called grumpy last year. I got exasperated, and I was never grumpy before on Twiv, and I think that was probably in reaction to things happening. Not the sadness of it all, but the scientific frustration at seeing incorrect conclusions made, incorrect statements made, and so forth. I felt exasperated, and I often took it out on Twiv, right? That's my punching bag. I don't make ships in bottles. No, I do Twiv, and I write blog posts, and I do other podcasts, and now I'm doing the incubator. That's what I do. But I am very frustrated at the fragmented nature of the science communication. It's never going to be all one source, of course, but there's just so much bad information going around. So that's why we do this, to try and make it better. We're not perfect, folks, but I think we give you the science. We tell you what's going on, and I tell you when it's my opinion, and you can do what you want. I think this is a good way to put it. Vince puts it where you can get at it. I think so. I think that's what I try to do. And when I read a paper, folks, I say, how can I explain this so people listening to me understand it? And I often say, I got to make a 15-minute video about it, but until I have the incubator up and running, I'm not able to do that because I have too many other things pulling at me. But I'm always driven by explaining it to you. So you hear, and people who listen to the pods and read my blogs and so forth. Thoughts on links between long COVID, ME/CFS, and persistent infections like EBV and others with similar symptoms being reactivated. So I don't think there's any evidence that there's virus in long COVID. I think this is a true acute infection. You get infected and the virus is gone in at least no more than a couple of weeks. Now, we thought the same for Ebola, and we were wrong. But we don't have those data yet. I don't think this virus integrates at all into your DNA. So I don't think there's any reactivation. I think the long COVID is a consequence of immune imbalance that's still not right, and we have to figure out how to set it right. I flew United, which is my airline that I have all my miles on, and which has a hub here at Newark. But I have to tell you, this morning I took the shuttle from the hotel to the airport, and of course there were lots of flight attendants. And I saw a certain airline that you're familiar with, and I thought of asking them if they knew a certain person, but I didn't, because I'm sure there are thousands of flight attendants at that particular airline. I'm being cryptic because I don't want to reveal anything. Yeah, I fly United. But because I have a lot of miles, right? And I get perks and stuff, which is makes it very comfortable. What BSL should hospitals have to handle pandemics? They don't need a laboratory of any BSL sort. They just need to have infection control practices in their patient areas, right? In the ICUs and so forth. They have to have patient control practices in place where patients are isolated, where PPE is donned before anyone goes in the room, right? And that sort of thing. And that they figured out very quickly. But the BSL, the biosafety laboratory, biosafety level, that applies to laboratories doing research. So, a medical center has labs and a hospital, right? But there are places that are just hospitals, obviously. They don't need to have BSL laboratories. Oh, H2, I was in your neck of the woods. If a fully vaccinated person got ill from another pathogen with similar symptoms and tested positive, would they be at risk of misdiagnosis and risk of mistreatment? Well, if they test positive for COVID-19, they probably have the infection, right? But they could be co-infected with something else. And so that could be overlooked, yes. So co-infections are often hard, and you'd have to have a very astute clinician, I think, who could pick up, there's something not quite COVID about this, and figure it out. And, in their and Daniel often said that, many of the patients, the severe patients, COVID patients, get secondary bacterial infections, for example. So yes, it's a real issue. Is there a test that can tell if someone had a natural infection a year or more ago? Yeah, for sure. You can look for antibodies against the viral nucleocapsid protein, which is not included in many of the vaccines, not all of them. The mRNA vaccines are spike only. The adenovirus vectored are spike only. So if you had a natural infection, you would look for antibodies to nucleocapsid. They would still be there. They'd be low in your serum, but they would not be zero. Low, to quote Daniel, low but not zero, right? And you could look for them, and those last for years. So you can really do historical surveys. And after SARS-1, they looked at people in the countryside in China, in the areas where they found the bats harboring them, and they found antibodies years later in the serum of those people against those viruses. Any progress on an oral COVID? So there's multiple ones where I made a list of them here for some other reason. Where is that? Do I have it here? I lost it. Well, there are lots of nasal sprays, but they're really early on. And the mucosal one you're thinking of it's an adenovirus, which you put in a capsule, which passes through your stomach, right, and is released in the intestines, and it replicates in the gut mucosa, but that will immunize other mucosas as well. I don't think this is going to be around for the pandemic. I think it's going to take too long. I could be wrong. Thank you, H. Sue, for your contribution. Really appreciate it. CDC RSV dashboard shows RSV cases above the 2019 peak. Any reason for this from a dynamics perspective? I think we have a larger population of susceptibles, right? Built up because kids were distanced for a long time. And I think also that it's spread in a different way. I don't know if masking... I saw a study recently where respiratory droplets didn't seem to be a main mechanism of RSV spread. So I think there are a bunch of explanations for that. Eileen is happy to be a booster for TWIV. Thank you. We'll always take boosters, yes, because they do work. They pay the bills. Thank you all. Above New Mexico, thank you for your contribution. I am PID and 2X Pfizer. I'm following the Ron Fouché approach, going mask-free, expecting minor infection for added immunity. Okay. I hope you're okay. I think you should be. You answered my question about keeping kids safe. You said wear a mask. Did you mean around the kids or around others? By the way, I noticed I passed 9:30. I will go to 10 o'clock tonight. I have to get up early. I have to catch a 7:20 train because I have a 9 a.m. call and I have to be in my office. So that means I have to get up at 5. And I got up at 4:30 the last 2 days in a row. I'm not complaining. It's just I'm tired. Maybe that's why I'm a little cranky. About boosters, right? You said wear a mask. Did you mean around the kids or around others? You're talking about in school, wear a mask in school. I forgot what you asked, actually. But you should wear it around people whose vaccination status you're not sure of, including school, right? Boosters for cancer patients. If you have an immunosuppressive state, which many cancer patients do, for example, getting an antibody to B cells will immunosuppress you. Then you have to ask your doctor because that population hasn't been trialed yet. You know, it's the kidney transplant population. But my suspicion is you could get a booster in that population. Yes, for sure. TWIV booster would be a great T-shirt. All right, Mark, that's great. I got to write that down. That's the one booster I do approve of. And by the way, doesn't matter what I approve of. I just tell you what I think. What did he say? I put it where you can reach it. What is it? TWIV booster. What about the graphics? We have any good graphics people that can make a nice graphic for that? I'd love to have that. I would pay you to do that. Yeah, the booster comments about TWIV are great. I'm not sure reasonably smart people understand what translation means. Protein synthesis. When we say translation, protein synthesis. Novavax works on Delta, but no go. We don't have anything on Novavax. It's too bad because they finished their phase 3, right? How quickly can boostering be ramped up? Well, if you're going to use the same vaccines, we're making them. We're cranking them out, so we'll be ready. It's not a problem. Events that are asking for proof of vaccination should allow people who have already had COVID. I don't see an immunological difference. Well, there is a slight immunological difference in that when you get infected and recover and then get a single dose of vaccine, you are better in terms of breadth of antibody response than anybody else, even twice vaccinated. So it improves what you already have. So that's a reason that you should be vaccinated. And, and there's a lot of heterogeneity in infections, right? So you could have a mild infection, maybe not have as strong an immune response as a more severe infection and so forth. So that heterogeneity doesn't exist in the vaccine. We're giving you the same dose of vaccine, right? So that's another reason why they may not be equivalent. Why aren't we shipping the supply of boosters to vaccinate other countries? We are, but it's not enough. And in fact, I read a great paper today modeling the effect of wealthy countries giving vaccines to less wealthy countries and how it would really help many aspects of the pandemic. But it's not happening enough. I agree with you. I think we should make as much as we can and give it away, for sure. Gamma is Brazilian, so the South African is beta. Yeah. Yep. Fabric versus paper. Well, in high frequency situations, like on an airplane, I wear two. I wear a surgical mask with a cloth mask on top of it. And depending on the mask, so an N95 will mainly protect you, right? And other masks will protect you and others, especially if they're worn properly. You don't have to wash the fabric every day, but multiple times a week. That is my practice. What's the main difference between the Pfizer and Moderna? So it's the same mRNA. It is a slightly different lipid nanoparticle formulation. The amount injected is different in terms of micrograms, and the Moderna second dose is a week later than the Pfizer. But essentially very similar. Is there any evidence of vaccinated people transmitting disease? Well, transmitting infection, right? I haven't seen any good information on that. People claim it, but I don't see it. Now, it's not going to be zero, but I do think the vaccine immunity and natural immunity to a certain extent after infection knocks down replication in the upper tract enough so that you're not going to transmit. You don't you need to make a lot of virus to transmit. And I think the vaccine knocks it down quite a bit. So those studies are underway, but I'd be surprised if they show extensive transmission. Yeah, we have another question the same. Can vaccinated people infect others? No good data on that. Let's see this. After 10 plus known close contact exposures, never having symptoms or positive test, I went and got a semi-quantitative antibody test hoping I'm positive. So, not everybody transmits who is infected. So 80% of transmissions are done by 20% of people, infected people. So maybe those people weren't the ones that are transmitting. It's possible. But 10 plus, that's at least one of them might be a transmitter. You'll see. Can adults carry latent RSV and give it to children? No, RSV doesn't go latent. But the vaccine can reactivate, Epstein-Barr and herpes virus infections, but not RSV. That virus does not do that, no. What do you think about New Zealand's reaction to the one COVID case? I always said New Zealand was going to have COVID again after they had none because it's very hard to keep out. I don't think you have to lock down the whole country for one case. I think there are other things that you can do. Maybe they can't do them all. Vaccinate, mask, so forth, distance. I don't think it really needed to be done, but I'm not in charge. Look, all I want is a meter that tells me what my immune system is doing in relation to SARS. I don't care about infection. Am I ready to fight off the possibility of getting sick? Well, that would be a correlative protection. So you could get your antibodies measured, and if they fall within the 20% level as shown in that paper that I just mentioned, then you're going to be protected against COVID. So you could do that. So you could go get an antibody test. CNN is pulled from the airports. It was on today. I can't get away from it. Do you think the U.S. will ever have a universal system for compiling medical data? No. I have no faith that this is not a country. It's 50 different countries, and that's the problem, right? So it's not happening, in my opinion. Not happening, but I'm just a cynic, right? Should my KN95 mask protect me? KN95 is very good at protecting you, but it's not 100%. You have to have some distance as well, unfortunately. You're vaxxed? That's probably the other part that the mask is not going to cover. Yeah, for sure. My favorite is Bill Hazeltine's claim that we'll have pandemic waves for 10 plus years and masks are here until 2032. Yeah, I don't think that's happening. Hazeltine, what does he write? A column for Wall Street Journal or Forbes, one of those? He's just off. I'm sorry. He's just off. I know Hazeltine. Used to be a virologist. Is it true that once You're a virologist always? Hmm. Why are there 2 shots for shingles and 3 for Hep B and 2 for Hep A? That's how they were trialed. Probably preclinical animal studies suggested you need multiple shots, and so then they trialed them, and they keep it like that in people. Yes, you do a small trial on every remake of flu vax. Absolutely. What criteria would be necessary to justify a booster update for SARS-CoV-2 similar to flu if disease increases? So for flu, they know how antibodies correlate with disease because they've studied it for 20, 30 years, right? So they say when neutralization by serum drops below this point, we're going to get more severe flu. We don't have that for SARS-CoV-2, all right? Do you know what I mean? It means you don't have to actually see disease increase. You just look at the antibody levels in serum of people. And since we know the correlation between that and severe disease, we can make that decision. We don't have that correlation with SARS-CoV-2. And so that's why we can't just look at antibody levels, although that's apparently what the CDC wants to do, and which I object to. Will the virus run out of mutation options? Not sure how to phrase it. That's a good way to put it. I like that. No, there are lots of ways a virus can mutate. We have 30,000 bases. That's 4 to the 30,000 different combinations. And even if you throw away half of them, that's still 4 to the 15,000 different combinations of bases. So it's got a lot of space. Now, it won't occupy it all, but no, it's as long as we throw selection at it, it will be selected. Absolutely. Awakenings about people with encephalitis lethargica linked circumstantially. Yeah. With the 1918 flu. Could fully recovered COVID-19 patients have health problems years later? Well, yes. That's what we're thinking. That's what we're worried about. But we don't have a long view of it yet, right? We've only been studying it for a year. So that's why it's so scary. Because it could go on a long time. Absolutely. Could a third dose induce longer lasting immunity due to a longer time? I don't think there's any evidence that a third dose gives you anything better after the second dose, at least for the B cell and T cell memory cells. Fully vaccinated mom still nursing my 14-month-old. How much protection do antibodies and breast milk provide? How long will we have antibodies after I stop nursing? So they're very good. If you have antibodies, then your child will have very good mucosal protection, but for a few months at the most. They don't last that long. Have there been other vaccine studies that look at the length of T and B cell duration? Yes, and I've been collecting papers, and I hope to do a TWIV episode on those, compare other vaccines. But for some of the other vaccines which have virus in the blood phase, like polio and measles, really decades-long duration of memory. It's amazing. It really is amazing. Rate of COVID has dropped dramatically in India. It's still less than 10% fully vaccinated. I don't think that's correct. Fully vaccinated, maybe. Yeah, they got 400 plus million people with at least one dose. Why? Because the virus ran out of susceptibles. It exhausted the susceptibles that were immediately available. But obviously there's still susceptibles around, but they were just not accessible to the virus. There will be another round, for sure, until they get the vaccination up. Absolutely. Monster, yes, Monster, right, with the 3 claw marks on it. I don't do any of those things. I don't know what they are. I just know bull thing, Red Bull. Here in the famous Faroe Islands, for immune memory, the nurses aspirate when injecting. This was discussed with the blood clots. Is it possible to do harm if the vaccine is injected? Yeah, the idea was if you put the vaccine in a blood vessel, this could induce clotting or other issues. So, but most people don't aspirate. Monster. I'm just getting to monster. Oh, wow. Let's get down here. Let's get down here a little quicker. Yeah, Moderna, I'm a bit better than Pfizer, but a bit is just not enough to convince me, right? How is infectious dose determined? It's not determined. We don't know what the infectious dose is. Absolutely. What kind of trials have been done on vaccinating new moms who are nursing? So they were excluded from the original trials, but some women did get pregnant during the trial, and so they were part of the study. But as far as nursing goes, I don't remember offhand. I think we have that data, but I just don't remember it. How do we assess T-cell function? There are no rapid T-cell function tests. There are rapid tests, say if you have virus T-cells, virus-specific T-cells. But function is hard. Function means the T-cell has to make cytokines or be a cytotoxic T-cell, and those are typically done in laboratories, experimental laboratories like Shane Crotty and Alessandro Sette. It's funny that it takes so long to get to some of the questions. So Lex told me that, Rogan drinks and smokes on the show, and he does other things as well. So that's not my shtick. It's fine. It works for him because he has a lot of people following him, right? But I like... I don't do that, and I don't know if I could talk to you people properly if I did that as well. So it's fine. It's good for him. It's his shtick. My shtick is something different. Look, let me tell you what Lex said. He said, I'm doing things that nobody else is doing. He said, you're pioneering education on YouTube, lectures and other modalities to the extent that nobody else is doing. Yeah, people do one-offs here and there, but I'm really consistent. I am persistent, not consistent. Persistent. I just keep doing it, right? So I'm pleased that he saw that. He really loved our talk. He said this is brilliant. He loved it, etc. So I'm happy he liked me, and hopefully we have a friendship and we stay in touch, because I think he's going to be a real player in the robotics world for sure. No doubt about that. Okay. Let's see what else we have here. How is COVID not going to be endemic? It is going to be endemic for sure. It's not going away. It is it's going to stay in people, and it is because they're always going to be susceptibles, and the virus will always circulate just like influenza does, and then it's in animals, and so they'll give it back to us. So it's with us forever, sadly. It's too bad. I really wish we could have not done that, but it's really a generalist. It can affect so many animals. Thank you, Michael, for your contribution. What would be the benefits of daily rapid antigen testing? So the advantage is that you test a kid before they leave home, right? And then you could say, oh, you're positive. You shouldn't go in today. And then you test them the next day. And if they're negative, then maybe you send them in. But you have to do really a third test to make sure. So that's the value. You don't get a viral load. No. You just know that the virus is there. You don't know how much of it is there. You don't know if they're transmitting, especially if they're vaccinated. You don't know if they're transmitting. So the only value is that you can keep them home and not potentially infect others. Yeah, let's revisit this random biological event. That's what life is. Make the most of what you've been given, okay? Ronnie, thank you for your contribution. You watched every lecture since 2016. Wow, that's impressive. Not many people have done that, right? Really appreciate it. There's no evidence about boosting, but have there been studies to boost people? Well, it's being done, right? Apparently 1 million people have gotten a third shot in the U.S. And I'm just saying there needs to be a basis for boosting, and there isn't any. What's the basis? Show me that the vaccine's not working. That's the point. It is working. If there were evidence connecting boosters with lowering the risk of long COVID, would you support them? Yeah, if you have that evidence, sure, absolutely. I'm not blanket against it. I'm just saying I don't have the data to say that they're necessary at this point. Let's see what. We're at 9:55. Let me see what. Let me thank all of the contributors. Thank you, Mark, for your contribution. Really appreciate it. Thank you, Sir Luke. Really appreciate it. Had COVID in March. Should I get a shot? Yeah, you should get one, Sir Luke. Your immunity would be better than most. It would be really great. Michelle, thank you for your contribution. Really appreciate it. Boy, a lot of comments on on Rogan. Some comments every week touch a nerve, right? It's really funny. Booster of the Month Club. That's funny. Don't you think it's crazy? It's unfortunate, and we will have them by the end of the year because they're being tested. But yeah, that's another piece of the puzzle, right? Absolutely cannot wait for that to happen. I'm looking for some more. So many questions yet left, folks. I know there's still 692 of you. I'm just amazed at your persistence. I'm amazed. You're still here. Thank you, Venti's Hidden Central Milker, for your contribution. Is Muller's ratchet and error threshold relevant to this virus? Oh, yes, of course. Muller's ratchet is restricting population diversity, and in any situation where that happens, it will make the virus less fit. I haven't seen that in the spread of SARS-CoV-2. The error threshold, this virus is reproducing very close to its error threshold. And if you increase this mutation rate 2 or 3-fold, it would extinct it. And we can do that with antivirals, in fact. And some of those are in testing, for sure. Here we go with Mike and Lori. Thank you for your contribution. Really appreciate it. I'm so excited to get working in the studio, the incubator. Credulopathic, thank you so much. Downside of too many boosters. I think I remember Brienne saying a few tweets ago that keeping antibodies so high could cause its own problems. Yeah, that's a theoretical issue, but I don't think they stay high for very long. You know, they decline every month about 2 to 3%. So I don't think that's an issue, frankly. But it's, yeah, I mean, the body naturally reduces antibody levels after any infection or vaccination. There's a reason for that, right? There are a lot of good questions here, which I'm really sad to skip over, but I have to thank the contributors and wrap this up. Bill, thank you so much. Thanks for staying grounded in science. Given the intensity of the outbreak, people have had COVID with no symptoms. The vaccination level, do studies suggest this outbreak will peak relatively soon? Well, as I said, and you need to have a 70 to 80% of the vaccine, the population immune in some way. And if you can't get that, you'll keep having outbreaks. When is your online class starting? In September. I'm planning to start it in September. So this is my online virology class. I'm going to teach introductory virology in the fall. I'm going to live stream it here on YouTube, hour and time to be determined. And I want to do it from the incubator. And I'm probably going to do it in the morning so that people in Europe can do it. The other hemisphere is a problem. I don't know what to do about that. How did deer become infected? I think via rodents, most likely, because rodents are probably infected as well. I don't think there's a lot of contact between humans and deer. I have a ton of deer in my backyard, and they run away from me. I can't get closer than 6 feet, so I don't think they would get it from people. Does the fact the labs work on coronas in a tube no, it doesn't worry me, and we can talk about this another time. They don't do that anymore. But as I said, it doesn't matter because they didn't have a virus anything close to this. So if they were working with it at BSL 0, it doesn't really matter because they didn't have the virus. It's a moot point, as they say, right? Thank you, Elise, for your contribution. I think your virology course would be a much better pre-med requirement than organic. I agree. I think that undergrads should have to take virology. And the problem is that not every university has someone who can teach it. I get emails all the time from people who suddenly have to teach virology at their university. They're not virologists, and so they're going to use my course to bone up and use my slides. No problem. You can do that. Thank you, Ian, for your contribution. Yeah, we passed 90,000 YouTube last week. We got to get the Patreon up. Yeah. It would help if I wrote people and thanked them. I have to do that. Thank you, Ralph, for... you're not silly. It's fine. It's absolutely fine. And thank you, Mark, for your contribution a little towards the incubator space. Yes, every little bit helps. And Mark did it twice, I think, unless I messed up with the clicking. Sylvia, thank you. The recent Josh Rogan piece on the lab leak. Well, look, there's no support for a lab leak. Rogan is wrong. I'm happy to go on and set it straight if you think that'll make a difference. A lot of you don't think I should do it. Thank you, Jamito. Oh, yeah, it's fine. I was kind of tongue-in-cheek. I always... Look, you guys wanted an epidemiologist. I got one on, right? I do the right thing eventually. A little stubborn, right? What the hell? I'm human. Thank you, Callista. See question about mom above. There's... Oh, here we go. Callista. Okay. My 86-year-old vax mom had back surgery, is in assisted living. Two workers tested positive. Mom has to quarantine for 2 weeks in facilities room. I don't think that's... So she has a potential exposure, but she's fully vaccinated. It's their policy. But she could just be tested for the first 3 or 4 days, and that would eliminate infection, I think. So, yes, it's unfortunate that... But would she be released from the facility, and this is keeping it from being released? Yeah, it's not great. Thank you for your contribution. TWIV Booster T-shirts. Thank you, Overjoyed. I appreciate it. You can make a graphic for the TWIV Booster T-shirt. Thanks. I'd love it. I'd make it. Thank you, my 310 Toyuma, for your contribution. Really appreciate it. Thank you, Neva. Good to see you. I wasn't too far from you. I was in Austin this week. Just overnight. Thank you, Jeff, for your contribution. Really appreciate it. I'm going to be on Jeff's pod in a couple of weeks. If you want me on your pod, just ask. If you want me to do anything related to viruses, I'm happy to help out. Just ask. Vincent at microbe.tv. Thank you, Jeffrey. 9 years of science education. I've had over 40. But I'm still learning. Thank you for your contribution. Livestream is a super resource. What the result of the incubator will be? The incubator is going to be a studio where I'll spend all my time recording science programs. podcasts, videos. I'm thinking of a 15-minute infectious disease daily, right? Every day. Would that be interesting? 15 minutes. What's going on in the world of infectious diseases? Anyway, I'm tossing through a lot of options for that. And it's the next phase of my career, right? It's dedicated, and I hope to raise money. Jeff is helping me to raise money. He's sometimes here. And then I can hire people to help me. Now, as you may know, Lex Friedman makes a lot of money on his pod because he has over 1 million downloads per episode. And boy, he can hire people. I don't want to run ads. I want to get donations and run it on donations because I think people will appreciate that we're trying to educate and help us to do that, especially if it's tax deductible, right? Okay, hopefully that explains it. Thank you, Barbara, for your contribution. Really appreciate it. And I wanted to thank our moderators. Right, Frank here. Thank you so much for modding, and Vanity Nutrition, who is not showing up right here in this part of the chat, but thank you, Vanity Nutrition. Thank you, Steve, for your contribution. Really appreciate it. Oh man, I don't want to get up at 5 tomorrow. You want to hear me read ads? I used to do Drobo, Curiosity Stream, all kinds of stuff, but I don't want to do that. I want people to support it. I could run ads, but I think people would prefer to have not, and I don't do ads on YouTube, as you know. Okay, we've reached the end of the contributions. That's it for this episode. Thank you all for coming. We didn't break 700 today, but that's fine. We got 330 thumbs up. I appreciate your support. And this is cool. Twiv is my booster. There's lots of possibilities. See you all next week. I'm going to be on vacation next week. Can you believe it? I'm taking a week off, but I'll still do this live stream. I'm still going to record Twiv. So I will see you next week. Have a good week, folks. Thanks for coming, and please be safe.